So in eukaryotic chromosomes, the ends of the chromosomes are called telomeres, and they get this name because they are just repeating sequences over and over and over again. So if I look at this example here, it's GGG, TTA, GGG, TTA, GGG, TTA, GGG, TTA, over and over and over and over again, hundreds of times. So it's repetitive sequence, and it's called non-coding sequence. It doesn't carry any information. So it's not going to be read by the cell to make anything. But it serves a very important function, and that function is to protect the ends of the chromosome. So why do the chromosome ends need to be protected? Well, it comes back to the way replication occurs. So let me just draw my little bubble here. So again, I have my origin, and then I have this peeled open DNA. So let's see, putting in some directionality here. So remember that every piece of DNA is going to have to start with a primer. So I was doing green for my primer. So I'll start down here. This is my primer, and then I'll elongate that. And I can go, and this is 5' to 3', because remember this is anti-parallel, and I can make a copy just fine all the way to the end of the chromosome, no problem in the leading strand. So in this example, this is the leading strand. So up here, it's going to be the lagging strand. So every Okazaki fragment is going to have to start with a primer. So let me start with a primer and then grow my new piece of DNA. There's my 5' and 3' end, and that's all honky-dory so far. And then let me put in my primer here. So primer has to match up against the DNA strand too. So I can put it all the way to the end and then fill in here. Okay, no worries yet. Now I try and replace my primers. So I'm going to chew out my primer. So I'm going to chew that out, and then I'm going to fill in a new piece of DNA. No problem there. Okay, but now if I chew this out, do I have any place to add on to? Is there something I can add on to fill in? No, because there's no more to the chromosome, right? So when I chew out this primer, I'm left with a little bit of a gap. My new piece of DNA is a tiny bit shorter than my old piece of DNA. And so what happens is that this gets chewed off to make the two pieces match. So now my new piece of DNA is a little bit shorter. Than my original piece of DNA. So the ends of chromosomes are continually getting a little bit shorter. This one will get shorter too, just on the other side, right? So every copy is getting a tiny bit shorter every time the cell copies its DNA and divides. So telomeres are there. Remember we said that that is repetitive, and doesn't carry information. So it's there to get eroded. So it erodes with no consequences. Right? If it doesn't have information, if I lose it, it doesn't matter. So as long as your telomeres are there, you can always lose a little bit of your chromosome and not have any consequence to the organism. But imagine something like an egg cell, right? So you're going to have a baby, you have this egg that came from your cells. Would you want those telomeres to be shorter than the telomeres that you had when you were a fertilized egg? Imagine if that was the case, if the telomeres that you gave to your offspring were always shorter than the ones you got from your parents. What would eventually happen is there would be no more telomeres. That means that the erosion would start to be in our actual useful DNA or DNA with information in it. And if that started to happen, it could hurt the organism. So in certain cells, we have an enzyme called telomerase. And telomerase lengthens telomeres. So this is especially important in cells called germline cells. Germline cells become eggs and sperm. So telomerase lengthens the telomeres in eggs and sperm, in the cells that become eggs and sperm, to make sure that offspring always get telomeres that are at least as long as the ones that the parent first got. Telomeres are really fascinating to me because they have a really interesting connection to aging and cancer. It turns out that our body cells have a very limited lifespan. So imagine if I took a skin cell from you and compared that skin cell to a skin cell that I got from a baby. The baby's skin cells would divide 80 times about in lab conditions, and then they would just stop. They won't divide anymore. They go into something called senescence. It's just kind of this quiet, resting state where they're alive, but they won't divide anymore. If I take those same cells from somebody who's older, like not an infant anymore, those same types of cells, they'll grow and divide for a while, but way less than the cells that came from a baby, only 10 or 20 times before they become senescent and no longer divide. And so this suggests that cells have a sort of way to keep track of their age, or the way to know how long that organism has been around, separate from the rest of your body. So you probably know, your heart gets old, your liver gets old, your different parts get old, but we think of those as structural parts going old. This tells us that separate from your organisms, we took these cells out of the person, right, and just grew them on plastic. They still knew that they were from an older organism, and that the way that they know is by telomere length. Cells will stop dividing if the telomeres get too short. So it won't let the cells divide anymore if the telomeres get too short. So that protects the information that's in our DNA. On the negative side, it also causes a lot of the symptoms that we associate with aging, or the nature of aging, which is that our cells don't divide as much anymore and the tissues don't renew themselves, and eventually they're just not functioning tissues anymore. And so what this really says is there's not really any way to live forever. Even if we cured every disease, our cells still know that they're older and that they can't keep dividing anymore. And so it's just it's obviously something that people are really interested in understanding, aging. And for example, they've found that in families that tend to live longer, they're actually genetically have a longer telomeres. The other thing that really excited scientists at first was the idea that maybe we could make our telomeres be longer artificially, like take a pill that would make our telomeres be longer, and then maybe we could live for a really, really long time, much longer than we do. But what we realized is that telomerase also has a dark side. And they've found that in 90% of cancers, telomerase is present at high levels. So telomerase seems to promote cancer. And the reason why is because cancer is uncontrolled cell division. Telomeres are there to stop cells from dividing when they've divided enough, right? Cancer is when cells just keep dividing when they shouldn't. And so one of the ways that they achieve this is by making telomerase, so that it overcomes this short telomere protection that our cells normally have. So again, that means that in cancer cells, the telomeres don't get shortened, and so they keep dividing even when they shouldn't divide anymore. And so probably a telomerase pill would not be a good idea because it would promote cancer. And if you had any sort of precancerous cells, it might cause those precancerous cells to become cancerous. And so telomerase did not turn out to be the fountain of youth, as people had thought. And there's probably a good reason why we've evolved this mechanism of having telomeres slow down our cell division and eventually stop cell division.